Okay, so this is going to be my week 10 review. So I normally do my weekly reviews on Friday at 11, and I didn't last week, and I can't this week or next week because I will be at a wedding and then doing papers with my students. Um, so I will be doing recordings like this. This time I didn't have a lot of time, so I'm just going to talk about protein synthesis, and then we'll talk about GMOs and evolution and everything we talk through next week for the next one that I do. Um, so this is kind of a little bit of a tricky one. There's a lot of details to it, so don't freak yourself out. You do need to understand it, but not in as much detail as I guess there is that you could know. Um, One thing I'd recommend doing is I'd recommend watching this YouTube video. It's one of the crash course videos that I found. They're really good. It does go into more detail and have some terminology that you don't even need to know, but at the same time, it helps explain it in a good way. And he's really quirky, so it's kind of fun to listen to him, I guess. So I would recommend watching that if you don't understand this at all. So I'm just going to go over really fast a little bit of terminology that you need to know and remember for this. So the first thing that you need to understand is so basically we're talking about protein synthesis. So this is taking a gene and turning it into a protein. And proteins code for lots of different things in our bodies. They have specific functions, and they're very important to us. So this is how we take a gene and we go through this process to produce a protein. So there are a few steps. So these are just some kind of terminology. The first thing, I put it down here a little bit, but just so you remember, DNA consists of adenine and thymine paired together, and then guanine and cytosine paired together, and it is double-stranded. So just remember DNA, D stands for double-stranded. That's a good way to remember it. So that is important to understand. We also have to deal with RNA, and we have a few types of RNA as well, but I'll talk about those in a minute. But the first thing I want you to understand is that RNA, instead of thymine, it has this base called uracil. So that will take the place of T. So if you have an A that needs to be paired with something and it's supposed to be RNA, instead of it being thymine, it will be uracil. So just watch out for that. But G and C paired are the exact same. And this is a single-stranded molecule, so a single-stranded section of genetic information. Okay, so I'm going to go up here really fast, then we'll get back down to these types of RNA. So the first thing is a promoter. So promoters are basically a sequence of nucleotide bases where the RNA polymerase binds to start transcription. So if you remember, so like a 3-pair nucleotide base sequence would be something like this. So a codon, that is what all of these are. So a codon would be each of these 3 bases put together. And if you see over here, so like let's say you get TYR, you look over here and that's tyrosine. So that is the type of amino acid you would have if you had that type of pairing. For example, let's say AUG. So you have your first letter over here, so A. So I'm going to look at that. Okay, A, and then my second letter is going to be U. So it's going to be in this one here. Oh, AUG, and that stands for that. So now I'd go over here, and that would be what I'd be looking for. Okay, so this is how you would break down and find what amino acid comes from what codon. So keep that in mind. I'll talk about it again in a minute, but I wanted you to understand that that's a codon, are the 3 base pair sequences. A promoter is a sequence of those that typically that's like the starting place for all of this to begin. So the first thing that we'll talk about is transcription. So the promoter is those 3 bases where the polymerase, which is an enzyme, binds to start the process of transcription. So if you remember, we talked about how we have DNA polymerase and how that helps in DNA replication, it helps with base pairing, and it's an enzyme. When we're talking about protein synthesis, we still have, at the first step, you would still have DNA polymerase, but as you're getting into transcription, you would use this enzyme called RNA polymerase. So it's an enzyme that synthesizes messenger RNA from a DNA template, and it binds it to the promoter. So that's the beginning of a gene, and then it unzips the DNA helix and creates nucleotides of RNA that are complementary to the DNA strand. And I'm just telling you that right now. I will talk about it when there's a picture so you can see what I'm talking about. And again, that video would be very helpful with this. And then there's some other videos as well on the website that can help with that. You can even Google it and it'll help you picture it if you can see it. But I'm just putting these out here first so that when I talk about them later, you know what I'm saying. Okay, so then we talked about how you have RNA. So you have 3 different types of RNA in this process. The first one is mRNA, and that stands for messenger RNA. So basically, that's a copy of a DNA gene, and it's produced during transcription. So again, I'll show you with the pictures, but understand that that's what that is. rRNA stands for ribosomal RNA. So that is actually, so this process occurs in the nucleus, and then it travels outside the nucleus into a ribosome. So the ribosome is where translation takes place, and so that's the next step of protein synthesis. So it's very important that that's there. Our RNA doesn't really do anything, like it doesn't actually have any genetic material, but it makes it part of the ribosome, and then it helps bind to the mRNA when it comes through the molecule. And then as it goes through the ribosome, it attaches to tRNA, which is transfer RNA, and that actually carries the amino acid sequences to the correct places to code for this right protein. So that sounds really confusing, but like I said, kind of how I do this is I will just show you the main things you need to know from each step, and that's the main things that those do. So other than that, don't worry about it, and it will make more sense when I talk about it with the pictures. Also read in your book. Like, I know it can be kind of dry and hard to understand, but like if you read in your book, there's pictures, there's like different types of things that can help you understand it a little bit better. So I recommend doing that as well. Okay, the next thing, just kind of going back, we've already talked about these ones, but so we've talked about how genes are basically a sequence of DNA that codes for a protein. So that gets broken down basically, and a protein is a bunch of amino acids put together in different sequences. So that's why we have different proteins, is because there's different sequences of those amino acids. And the amino acids come from codons. So like all of these here put together, those will code for, so like that's one amino acid, that's one amino acid, that's one amino acid. And that's like a codon. So this codon would code for, like AUG would code for that, which you can see here, which we actually talked about just a minute ago. So just understand, so that's what that is. So if you understand those terminology, that will be most helpful when you're trying to go through the process. Okay, so protein synthesis, like I said, it consists of 2 parts. So you have transcription, which is basically copying the DNA into RNA. So it makes a transcript. So transcription, transcript, okay? So it's like copying like a book you need or something like that. So I think in one of the videos he talks about making Hot Pockets. So it would be like him breaking into the facility that has the recipes and then writing shorthand the recipe so it's not the exact same, but it's the same steps, and then taking that and then actually making the product, which would be like taking DNA to RNA to protein, but like instruction manual to machinery to Hot Pocket. It'll make sense if you watch the video, but it's actually a really cool process. So just understand that that's what that is. It's copying the DNA into an instruction manual. Translation is more like assembly of proteins. So it translates. So you get these things called codons, and they're like 3 sequence base pairs of like AUG or UUG or things like that. And to me, that's like a different language. So it has to translate. So it needs a translator, which is the tRNA actually, and it translates it into specific amino acids, which is when you would get like, so you can see right here, so it goes from DNA to RNA to protein. So that's called the central dogma. So when you get that, you get like these 3 base pairs would be a codon. So that would be what it would be you would translate that into this. So it's translating it from one thing to another. and a bunch of these amino acids put together is a polypeptide chain, and then that would make a protein. But yeah, so that's basically like the overflow of genetic information. So it goes from DNA to RNA to protein. So you can see right here, so DNA is double-stranded. So you would have this, then you would have that RNA polymerase come in, and it would go on top of it right here and kind of split it down the middle, and then it would just make a copy into RNA, but it would just be single-stranded. So it would be almost an exact copy of this bottom strand, but wherever there would be a T, there would be a U instead. I know this stuff seems way confusing. If you watch the video or like any type of like little like animation, it really makes a lot more sense because it's hard for me to explain like this. But if you need more help, come in and talk to me too. I'd be happy to. And then it goes from RNA, and then so actually at this point right here, this is where messenger RNA is. So the DNA to the RNA, that's actually mRNA right here. And then it goes into the ribosome right here, and that is where it would bind to the rRNA. And then as it's coming out of the ribosome, it also binds to the transfer RNA. And then the transfer RNA carries the amino acids to go where they need to. Like, okay, this one needs to go there, that one needs to go there. And then that codes for the protein. Okay, so again, the basic concept of this is called the central dogma. So again, it's going from DNA to protein, basically. So DNA, RNA to protein. So as you go through that, you can see here too, you have like mRNAs right here. There's the ribosome. The mRNA binds to the rRNA, which isn't listed, but that's what happens. And as it goes through, it produces that little strand of amino acids. So it's actually a really, really cool process. There's really cool animations. I'll try to find some and add them to the file as well. But keep that in mind that is basically the overview of it. If you understand that would be a codon, and that codons code for amino acids, and amino acids make up a polypeptide chain, which becomes a protein. That is really the basic overview of everything you need to know. So this basically is just another like example of the steps. And then this is basically just like a practical like example of it, I guess, like that. So like all the steps, but in like a real life scenario type of a thing. I'm not going to go over it. You can read it, and if that helps you, that's great. Again, the video is super helpful. Okay, and then this is actually something that you need to know. This is called like, basically this is a figure of the genetic code. So on like an exam or something like that, Dr. Peterson will be like, okay, so you have AA, U and AGU and GGT, and we'll ask you to give the amino acids that it corresponds with. So how you would do that is, okay, so if you know it's AA, so first letter, second letter, oh look, there's AAG or AAU, whatever I said, and then you would find that over there and write down that would be what it would be. So he would put a graph like this or pick this picture like on an exam, but you would need to know how to read it. So if that doesn't make sense, let me know. The third letter's over here as well, but typically when you find the first two, you can kind of figure it out. If that doesn't make sense to you, come in and I will work with you. Again, I'm sorry that I am not going to be there. And I will post another one where I will post just the PowerPoint, and then I'll also post like the lecture of me talking as well. And it will be longer because it will cover a lot more information. But it's kind of a concept that's tricky and a little bit difficult, but we don't talk about it a lot in the class. Like, that's why I kind of wanted to focus just on this, because it's a lot of information, but you really don't need to know that much of it. Again, if you understand central dogma, DNA, RNA to protein, and then understand those terms too, that would be my best advice for you. I hope you guys have a great weekend, and email me if you need help, work on your papers. Yeah, you guys should be great, and I will post another one with more of the other stuff later. I just didn't have a lot of time tonight, so I apologize. I hope this is helpful, and I will see you guys on Monday.